Okay, and now we are moving to the last part of our workshop today, and that is the panel session. So all the speakers of the day, please come and join us here. And Gaute will be moderating this session.
Okay, thank you. I have a few questions prepared here, and I think one, I mean, what we learned from, I guess, especially the talks of Clay and Nuno earlier today was these fantastic developments when it comes to like connectomics and like anatomy, really mapping out the connectomics, and Clay promised full mouse connectomics in like EM in a couple of years. That sounds good. So, but I'm also curious about how much can we, of course, if you want to make a model, you would like to know the anatomy, but ideally you'd also like to know something about the electrical properties of the membrane and so on. And how much of that can we dream of obtaining by extending the kind of studies that's already done? Anyone who can speak on that?
Yeah, first, I take no responsibility for Clay's promises of 2 years.
I take no responsibilities. Mine was meant to be, we did a hard thing. Here's another promise. Give us 20 years.
But to answer your other question, Gaute, I think that I think the path that is possible right now is that if you have all of those electrophysiologic properties from some other experiments, and you know that they correspond to a particular cell type, and you have the morphology of that cell type, now we can go to the EM and find the neurons that have the same morphology and link it through it. And I mean, that's the approach that we took with Costas. He generated biophysical models of layer 2-3 based on the Allen Institute layer 2-3 cells, and then we brought to that now the connectivity of those same cell types.
But say for the axons, like this complicated axon jungle that Clay showed, right? I mean, do you know how is it like a way to get an idea about sort of like the electric parameters there?
Nuno, do you want to field that question? I think let's go back to Terry's first slide of the different scales. You know, it's possible to, in our dreams, and again, this is in our dreams, you know, for the mouse brain, go from the smallest to the largest. But you got to get molecules and biophysics and other things in there too. And I think the glue that holds all that together will be cell types, and cell types defined probably a combination of transcriptomics, anatomy, and probably biophysics as well. And no experiment is going to have it all. And I might have gone through it quickly, Nuno might have gone through it slightly less quickly, but the idea of Rosetta Stones to link the various modalities together is key. And it's sort of the Allen Institute mantra in the past decade, and really in the next decade as well, that the Rosetta Stones that we're thinking about now are largely morphology, that you know, those beautiful morphologies that Nuno showed with all the different colors, that's from patch-seq experiments where you patch in a neuron, you do biophysics, you suck out the cell body, and you do transcriptomics on that, and you get three things. You get morphology, you get the transcriptome, and you get biophysics. And via the morphological Rosetta Stone, you can map that onto connectomics. So that's how you get from biophysics to connectomics. I think it might be easier. Well, no, everything's hard. It'll be slightly different with the promised but non-existent axonal connectome, where in the same experiment, one will be able to do anatomy with antibodies, and unlike with EM currently at least, you can get cell types either from antibodies or ultimately from RNA, from the transcriptome. So, from there, I think it would be a mistake. I'm sorry if I'm going on too long, but I think it'll be a mistake to do the promised projectome unless one has it linked to the transcriptional types. And then from there, you know what they are, and in the same kind of thing, you can do biophysics on those via another Rosetta Stone.
So,
Nuno,
may I honestly, I'm dying to know, from our single unit recordings, why only like a very selective subset of cells that are rhythmic. So we've been trying to figure out what can we do to see why those neurons are different. And you just said maybe that's possible to patch those neurons, look at their electrophysiological property, and then do some transcriptomics, and then you also have the connectome data, then maybe that would really help us to understand the network activity closer.
Yeah.
Can I jump in? So I think that, focusing on the biophysical properties locally, within the neuron, and focusing on neural types is incredibly important, but there's neglecting another biophysical property which I think has been neglected, which is time delays. And in fact, one of the hopes from the project that Clay discussed, which was the White Matter project, is that we'll have a much better idea of time delays between different pathways. And we know a few of them, like for example, thalamic feedback from the cortex, from layer 6 to the thalamus. There's 10 times more axons going down than going up, and they have time delays that range on an order of magnitude. And, and interestingly, very few people who build models worry about time delays, and especially in the cortex, the long range fibers, association fibers, have time delays of tens of milliseconds. And we now know, there's hints that people that were studying, for example, learning and looking at changes, everybody focuses on synapses, but people have seen changes in the white matter. So what is that telling us? It's telling us is that, well, we know that axons, biophysically, that the speed depends on the axon caliber and whether it's myelinated or not. And so you can imagine that that is also being, it's an intrinsic property that can also be regulated by learning. So
that is maybe something we can get from anatomy already, right? From what Clay showed, the thickness of axons.
We can, and we've in fact, I have developed a model of sleep spindles, which is driven by those observed 3 meter per second long range of myelinated fibers within the cortex. We showed recordings from ECOG that there's traveling waves, that circular traveling waves, and that can be reproduced very nicely in model with time delays. So I'm just saying that I really would like more focus put on time rather than just the local mechanisms.
Nuno, you were going to say something?
Yeah, I was just going to say that what Terry just brought up, it brings an important kind of point in this kind of cycle between electrophysiology and the anatomy, which is, the electron microscopy data sets have a lot of data there, but in a way we only extract what we think is important to extract. And so in this cycle, it's where I think the modeling plays a very important role. If there is some particular feature that the models are very sensitive to, those are kind of the features that we can then go back to the EM and maybe and segment first. So for example, we know that inhibitory cells, but also excitatory cells, they get myelinated for little bits of time. I mean, what is that for? Why is that bit of myelin? But we don't put that on the data that we extract, but we could. If that was thought to be important, is those kind of, does this kind of changes the phase of the input of something? If there's a theoretical aspect of which it should be valued, that will guide that extraction instead of other parts of the, so this interaction with modeling, I think is kind of crucial to explore this kind of microcosmos that this large AM data set allows us to see.
Okay, maybe I'd like then to go to another question, because I think another topic of today's talks was this, how, I mean, both Andreas and Terry showed examples where you actually use input from machine learning, these developments there, to learn about the brain. And I guess another approach there has been that people have sort of, I guess, compared the representations, neural representation in these deep networks trained for, say, image recognition, and they see these clear analogies between like different layers in their deep network and remember representations in the brain. So, but what are other, I mean, there must be lots of opportunities there for sort of taking advantage of machine learning, or you would think. So any ideas there, Andreas or Terry?
Yeah, just briefly, there's a tremendous number of different models. Deep learning has gotten a lot of attention, but there are other models. For example, GANs, generative adversary networks. What makes those attractive is that they're generative, whereas the feed-forward networks, although they're very powerful, they are just basically reflexes, input-output devices, which make a transformation, very sophisticated transformation. But if you can generate complex output patterns, you're a step closer to the brain, because one of the things we know about the brain is that it's generative. You don't need to have a stimulus in order to have your brain activated. There's spontaneous activity in the absence of sensory input. And that really has to put constraints on the modeling. And let me just add one more example, which is the hippocampus, which has been extremely well studied. And we see how it is driven by serendipity, the discovery of place cells and grid cells and so forth. But we still don't fully understand how it fits in computationally. Lots of ideas. But one of them that comes from the anatomy, just the way things are organized in terms of the inputs coming in from the cortex, entorhinal cortex, through the dentate gyrus, which looks like it's spreading out the information into a higher space, higher dimensional space, and then the CA3, which is a recurrent network, and then back through CA1. That looks like something called an autoencoder. And the purpose of an autoencoder is to extract latent variables in the inner loop, which is in the case of the CA3, that capture underlying higher order conceptual, concentrating all the information coming into a code that can allow you to recover the original input. That's what an autoencoder does. It just compresses it and then expands it. And I think that this, it gives us a different conceptual framework if we sort of think about these computational primitives and try to map them onto the anatomy, and then use that to predict for new experiments that could be done to test whether that network, that hippocampus or some cortical network actually does.
That also goes back to the work in the 60s with like David Marr and David Wilshaw, where they had very simple neural units, but they did computations based on anatomy connection, like associative memories and things like that.
Yeah, I think in fact, the more constraints we can put into the models, and coming now from the machine learning world, the more constraints we put into the models, the more we're going to get out of it in terms of being able to map it onto the actual complexity of the cortical circuitry.
I think one problem with modeling in general is that, I mean, you can think of deep learning as a tool to analyze data. It's just another statistical tool that you can analyze data. But in addition, it provides you a common framework to compare, to model the brain in terms of behavior, right? So if you have an idea of how this part of the brain works, you can use machine learning tools, from deep learning, in particular neural networks of different forms, to see how well you can recapitulate the behavior given the assumptions that you put in. Now, the problem becomes that if you take, for example, what was said earlier, let's say you take biophysical information, you try and build a model, then there's no, at least right now, because you don't have all the biophysical information. It's like you have too much details. And there's many ways, because you're always missing information, you can reparameterize the model with other nonlinearities. So it's not clear if that approach, if you're trying to, let's say, ultimately model behavior, which is what a lot of us are interested in, then by using more complexity, increasing the complexity of the model, there's no evidence right now that that is giving you a more faithful, predictive model of the behavior. And it sort of makes sense mathematically because now you have too many variables and you are missing critical variables, compensating with others. So I think this is the challenge when we're talking about bridging multiple scales of analysis. I mean, this is not just true in neuroscience. If you take physics, for example, where things are simpler in a way, it's very hard to go from, or organic chemistry, to go from the quantum level to describing chemical reactions, right? So, and so anyway, so that's kind of one challenge, if where, deep learning is not necessarily trying to increase complexity, but just improve performance. Now, the other one is, and it was alluded to it earlier, a lot of people think of deep learning as a black box, and in some ways it's not. And in fact, the deep learning community is very actively trying to develop tools to open this black box, to try and understand, for example, even if feed-forward CNN networks, what is the properties of that network that enable it to do object recognition under these conditions and fail under other conditions? Because there's a lot of failures of these models that are very dramatic, and like adversarial attacks, for example. Those that don't know, it's like if you train a network to say cats and dogs, can take a cat and change even a single pixel on the image that the human cannot even notice, and now the network is convinced it becomes a dog. So the community there is very active, is very successful, it has a lot of funding, it's developing a lot of tools to open these quote unquote black boxes. And I think that now that we have a common way to model the brain using those tools, we can, from a practical point of view, now use these tools to understand, because, neural network is just a much simpler form of a network that has activations, synapses, and it doesn't have dynamics. And if we cannot even understand how that works to our satisfaction as neuroscientists, then how are we going to understand the brain? So I think it's the right time for this cross-fertilization between these two fields.
So there is a path, a very interesting path to go along the lines that Andreas just outlined for us. So, these units that are used in deep learning are very primitive. They're rectified linear units. So there's a threshold, and above that it's linear. But we know that real neurons have all sorts of intrinsic properties that make them dynamical systems. Well, it turns out that it is possible to include intrinsic properties while you're optimizing. So backprop, it gives you a gradient, and you could use that to modify any parameter. And it could be ion occurrence, it could be thresholds, it could be times. For example, I didn't mention it, but in our network, we actually did modify time constants. And so by putting in more of the parameters to modify, it will be able to understand a little bit more about how they could be used to help the computation, and might even improve on the performance.
Anyone else want to chime in here? Okay, I have one more question. I think we have 5 more minutes, and this is sort of like, what biological mechanisms, if properly implemented in models, will lead to the most dramatic improvements in applications of models. And I guess if you look at these like biophysically detailed models like what Mikael talked about and Alan V1 model, they have obviously they have neurons and they have synapses and they have ion channels, but it's not so much. Even though I think the blue brain model has this short-term plasticity, but there's not so much, I think, learning rules and long-term plasticity. And I think one thing actually that was so about the work of BOA, that actually this, if you want to model we have done some work that you can model the swelling of neurons, which probably has to do with activity. I mean, it has to, it is like osmotic flow that actually sort of that is like, it could be an effect for why something that the neuron does actually affects how easily the CSF can flow and take the amyloids with it. So that's actually a way to do it. But otherwise, what is missing the most in the models we have today when we talk about at least the biophysical detail models, like more cell types or learning rules or neuromodulators or nonlinear
Exactly.
phenomena?
Yeah, I think it depends what you're aiming for. When you see what is missing, can you clarify, get what, prove
No,
what in the model?
that's like,
I think that makes a big difference on the answer.
yeah,
Like,
I'm a poet. I just ask questions, right? So I think you have to interpret the question as you.
any way we want.
Yeah, exactly.
Good questions are really important, and I think this is a good one.
Yeah.
Can I I've been playing poet today, so can we slightly invert the question?
Okay.
I think the biggest gift that those of us who collect data can give the modelers today is the diversity of cell types and the diversity of information about cell types. Do any modelers have ideas how to use that? You know, not one or two or four, but 50.
So John Doyle and I have an archive paper which explores that, and the title is it's in the realm of control theory, and it's specifically speed accuracy trade-offs, which are all over the place in motor systems. And what we showed is that you can, even though there are limitations on speed and accuracy in axons, so the big ones are fast, but they can't transmit information much, they have fewer spikes. But small ones, you can get a lot of them transmitting a lot of information, but much more slowly. But it turns out that if you have that diversity, which you see in almost all big nerves, then you get something that's called a diversity-enabled sweet spot, which gives you performance that looks as if it's fast and accurate, even though any individual component has limitations. And I think that that's going to be a general principle. It's not just for axons. It's going to be, diversity could well be one of the important overarching principles in all of biology. The fact that we have diversity within a species, diversity within a gene pool, diversity at every level, right? This has got to be important.
Absolutely.
Yeah, but I also think, I mean, at least when you make a model, I mean, the hard thing is really fitting the parameters like the conductances, right? And this is something that typically, like the channel conductances. And so this is typically, I mean, real brains can do this on their own because they have some homeostatic rules. So it just seems that if you had these homeostatic rules and time enough to run the model for a long time, this could save us a lot of pain in finding this like a sensible parameter set.
So why don't we put homeostatic rules in our models? And so that would automatically take care of a lot of the problems.
Do we know the homeostatic rules?
Well,
you know?
you know, it's been studied, Gina Torrigiano, for overall normalization of synaptic conductances on the neuron, and also Eve Marder has studied it in terms of her neurons and how they adapt. So I think there's already enough out there to actually begin to incorporate that.
And you have to run them for a long time, right? That's also a thing, right? That you want to like the models to run for like hours or whatever days of biological time, right? Which is sort of makes
well, you know, computers are getting faster.
Yeah. but not that much faster. Yeah, Stephanie?
One thing I'll add is the main thing where I think we're missing in the modeling field is that we just start mostly modeling neurons, right? And these neurons live in a brain and they live in a body and they live in a motivated or hungry or, you know, all these different things that we never think about in our model. We build our model and then we have some drives that we give to it and how it reacts. But those drives are really dependent on the body that we're living in and our muscles and our motivation. And we're just getting at the surface of how to put these models together, never mind how to drive them in the right way in a awake behaving, motivated, hungry body. You know, we're starting to build in vessels and glia and think about the interaction, but there's so much more that we have to think about when we're thinking about these networks working in real states and how that's going to happen in a real awake behaving being. And probably it has to do with certain neurons being driven at certain times and these ongoing gamma little generators, they may be tied to a certain cholinergic system. And the more we think in experiments where we can start to probe these different behavioral things might help us disambiguate what the different parts of the network are doing. But I would say that's one of the biggest things we're missing right now. You know, I'm working in the field of brain stimulation, and everything is about modeling the effect of the brain stimulation on the neurons. We don't talk about the vessels, we don't talk about the vascular, we don't talk about the skin. I mean, there's so many other things that, you know, I guess it's by necessity, we have to start simple, but I think we're getting so much better now with building these models that we need to start thinking about other non-neuronal system interactions.
Okay, maybe this is a good place to stop. It's 9:15 here, but I don't know what it is with your something else around the world. 9:15 something. So I think it's a good place to stop, and then we'll meet again tomorrow for the last day, I think, at the same time. Is there some messages that from the event team?
No, just thanks to everyone for a great second day. We're excited for one more tomorrow.
Thank you, everybody. Have a
Thank
nice
you, Ashley, and everyone. Good.
Okay, ha det bra.
See you tomorrow.
Take care.